Hello, everyone, and thanks for joining us. I'm Arslan Arif, publisher and founder of Endpoints News, and I'm joined today by the team at Agilex Biolabs to discuss leveraging APAC partnerships to de-risk clinical trials, how biotechs work with biolabs and CROs to manage timelines and success. From Agilex Biolabs, we have Julia Jones, Vice President, Business Development, and Dr. Kurt Sales, Director of Immunoassay. We're going to start with a presentation from Kurt, and both Kurt and Julia will take questions towards the end. And now, without further ado, I give you Dr. Kurt Sales. Kurt?
Thanks, Arslan, and hello, viewers. So, as Arslan mentioned, this small webinar is just basically how biotechs work with biolabs and CROs to manage timelines and success. And it really focuses on the decisions that need to be made in order to create a successful clinical trial. So over the past couple of webinars, we focused on the early phase and bringing your trials to Australia. And in this webinar, we thought we'd focus really around the key early decisions that really determine success, specifically around the biolabs and the assays that are needed for secondary and tertiary endpoint analysis for your clinical trial. And there really are two approaches that are key in order for us to get the trials successfully set up and to completion. And these really hinge around the pharmacokinetics, immunogenicity biomarker assays that are required for your clinical trial. And the two ways of approaching this is either a direct approach, which we call a biolabs first approach, or a CRO-led clinical program where the labs are contracted through the third-party CRO. In this webinar, we'll focus on some of those decisions and give you a flavor of exactly the process and the decisions that need to be made in order to make this successful. It's all about partnership and collaboration and just generally taking the assays off the critical path, reducing the amount of stress associated with the clinical trial, especially for new and emerging markets or where the client is in another country and the trial is being run in Australia. We'll touch a little bit on some rescue projects and processes around this. We've seen quite a lot of this coming through as the rest of the world is shut down still due to COVID, but small biotechs had started their clinical trial and this got put on hold because of COVID and shutdown. The trial then moved to Australia, and it's how we can then assist and rescue the assay and method development from that point onwards. So just as a reminder on the reasons why you've chosen Australia for those viewers who hadn't seen our previous webinars, and that's really because Australia has been conducting clinical trials now for over 25 years. And it's a really speedy, expedited process. A full CMC package isn't required in order to bring your trial to Australia, an IB, investigator brochure, which contains all of your supporting materials, insurance certificates, and really everything hinges around the ethics committee evaluating all of the information and making a decision. The decision is really made very rapidly. The ethics committees sit very regularly in order to evaluate the applications. And this really means that the timeliness and speed is in the favor of the client, especially small biotech companies that are in the sort of burning rather than earning phase of their life cycle. And this really enables the clinical trial to start really within 6 weeks after ethics committee submission. We have fantastic expertise. Most of our key opinion leaders have trained overseas, have fantastic international kudos and reputations, and we have a superb network of physicians that are able to be brought in for any disease indication or evaluation. Most importantly around the clinical trial is that the data that is generated is acceptable throughout the world. So all of the regulatory agencies acknowledge the quality of our work, and many of our small biotech companies that are doing their early phase within Australia go ahead and lodge their IND in the FDA or EMA. As a biolab, we qualify for the research and development rebate of 43.5%. This is an incentive put onto the Australian government, and really all of the activities associated with the clinical trial qualify for that rebate. And then just finally, with the global shutdown over the past year since the COVID-19 pandemic first reared itself, Australia has been open for business as usual throughout this entire time. We have COVID management plans in place, but we've really been unhindered and uninfected by the global pandemic, which has been a great benefit for many of our customers throughout the United States and Europe to bring their early phase studies to Australia in order to continue their work. So what are the key early decisions that really need to be made? So if we consider the biolab's first approach, this is where you, the small biotech company, might approach us directly, either through our website or through various campaigns that we've set up or launched. We're also on LinkedIn. And so this means that there are a variety of platforms that we can be contacted through in order to start those initial discussions. And what we'd like to do is to get an indication or be able to evaluate what the molecules look like. Most of what we deal with are novel biological entities or novel chemical entities, and they've never been done before. And so there's inherently a limitation around the types of reagents that are commercially available, either to build a ligand binding assay or for chromatographic assays. So the process is pretty quick and very simple. We put a confidentiality agreement in place. This can take as little as a couple of hours, a couple of emails backwards and forwards. We can then share information freely to get a flavor of your molecule, how you'd like to work, what you're interested in doing. Is it an early phase 1 trial, or have you already done some work on the molecule in another country, and this is going to be a patient study as a follow-on to your healthy volunteer work. Once we have all of the information and our scientists can evaluate the preclinical data, or whether it's a method transfer and you're bringing the assay in from another CRO or another biolab to us, we can then start drafting a process plan. And this is pretty simple. We can put a letter of intent in place to fast track the work. And this is just really a document that says, you're going to work with us, we're going to work with you, and we are happy to not invoice you for a certain period of time, up to a couple of months, to enable you to set up your local entity for the R&D tax, or for you to contract a CRO that ultimately would then wind our contract into the master contract agreement with yourselves for the clinical trial. A really important part of the process is enabling us to start the method development. And the front end of the method development is really any kind of procurement that might take place. So these would be specialty reagents, secondary antibodies, primary antibodies, antigens for capture detection, or deuterated internal standards for chromatography assays. And then to also get reagents from the client, generally the test item, or any other kind of specialty reagents required for getting the assay set up. This can take 4 to 6 weeks, depending on where the reagents are available globally and through which suppliers. One of the issues with the COVID has been the delay in procurement. There are fewer flights into and out of most countries, so inherently it takes a bit longer to get the procurement and test items into the laboratory and booked into the systems. But once we have those specialty reagents in, we can then commence the assay development. And I'll delve a little bit more deeply into that from a biolab perspective in the next couple of slides. But really, this sets in play a partnership with the customer to just de-risk that front-end part of the bioanalytical in order to take that off the critical path. As a biolab, we can recommend CROs if the customer hasn't already contacted a CRO to manage the trial. And we work with all of the leading CROs within Australia and globally. And as I mentioned before, we are registered for the R&D tax rebate. And so if the customer is earning less than $20 million revenue per annum, they would then qualify for the R&D tax rebate and all the activities that the biolab would then undertake. And we can then provide additional information on the CROs, the landscape, putting in contact with individuals to assist with getting an Australian entity, insurance up and running in Australia, and really enable an end-to-end process to just make the trial that much more seamless. The second approach really is what we call the CRO Led Clinical Program. And here it's where a CRO or third party will contact us on your behalf. They would put a CDA in place with themselves, or there might be a tripartite CDA in place to enable free discussion and exchange of information. And so the process is very similar to what I had described in the previous slide, except that we're now going through a third party. And inherently, this does take a little bit longer in terms of the process, as there's generally an intermediary, a project manager within the CRO that's managing your clinical trial and then contacting us as a biolab for the pharmacokinetics, immunogenicity, pharmacodynamic biomarker assays that might be required as secondary endpoint evaluations for the trial. Again, we can set up a letter of intent with the CRO. The CRO contracting generally takes a little bit longer, as it really encompasses the entirety of the clinical trial. And so we'd want to get started as soon as possible, again, to take the assay off the critical path. As you'll see as we move through the slides, method development validation is a process that does take a considerable amount of time, depending on the complexity of your molecule. More so for a biologic, and more so for a specialty NCE or NBE, where there aren't readily available, high quality, commercially available reagents. So study rescue is something that we're seeing a lot of, and we're actually doing quite a lot of study rescues. And these are studies that that either fell over in another country, for lack of another word, where the contracted lab was unable to develop and validate a method to the EMA or FDA bioanalytical guidances. There might be issues with selectivity, specificity. There might just be something inherently wrong with being able to try and get the drug tolerance and sensitivity right for an immunogenicity assay for a biologic, where the dosing is escalating, or where it's being dosed into a high concentrations within a patient population, or the clinical trial has come to a grinding halt in the country because of the COVID pandemic and the phase 1 units shutting down, and the sponsor has then made the decision to bring the assay over to Australia and set up shop here and conduct the clinical trial in Australia. And so under these circumstances, we can then transfer the method in, we can evaluate the methodology of the other lab, and with our high sensitive equipment and skilled workforce, we can easily identify the key issues with the assay, and we can re-optimize it so that it's fit for purpose. Most importantly is the long-term stability, because once a trial is started and we don't have an assay in place, we're constantly then playing catch up to get enough proven stability for the duration of the clinical trial. Ordinarily, we would prefer to have at least 1 month's proven stability at -80 and -20 degrees centigrade, ahead of first patient, first dose, as that just gives the assurance that the collecting conditions throughout the clinical trial are optimal, and that the integrity of the test item, once it's dosed into the subject, isn't compromised during the storage period. So we can get this set up pretty quickly. Again, it just takes a CDA, a briefing, and we can turn around a quote in a couple of days. We can put together a letter of intent, and once we have those critical reagents in place, we can easily start up a method development and validation. And these we would then do back-to-back, as well as establish from scratch a long-term stability program in order to start getting that very vital stability data for your clinical trial. So what does the Australian ecosystem look like? I've touched on this in previous webinars, but this slide really just gives you a flavor of all of the key players involved within the Australian ecosystem that are responsible for the clinical trial. So, initially focusing on the left-hand side of the slide, we have the biolabs and we have the specialty safety labs. We have the CROs, and we work with all of the major CROs throughout Australia and within the region. And each one of these CROs is responsible for setting up and the conduct of the clinical trial, including either the subcontracting of the laboratory portion for the safety study, and also for the pharmacokinetic, immunogenicity, and biomarker aspects of the assay. And then we have all of the other key players, the phase 1 units, the hospitals in the patient study, and the critical other services, such as the logistics providers and tax consultants for R&D rebate, and also third-party sort of toxicologists and specialists that might be required for a patient study or for a rare disease population study. So this sets in place the ecosystem on the slide of all the players that are key to the successful delivery of the clinical trial. And as a biolabs, Agilex Biolabs is really well placed within this ecosystem, because we work with everyone, and we've been around for 25 years with the largest and most extensive biolabs in terms of our expertise and knowledge. We can easily guide a new customer as to how to navigate through this complex ecosystem, and also put you in touch with the key CROs that could manage your project. So the key process elements, really in a linear fashion, are contact us, a preliminary call, we can review all of the documentation, and we can provide an assessment on the study, give an indication of timelines. We can easily put a master services agreement together, or a letter of intent ahead of the master services agreement. And this enables us to work in a very fluid way with the customer to get the method set up. And then once we have those key contracts in place, to start with, we can then procure material. We would procure material on your behalf, and it would be billed to the project as pass-through cost. And then we can start method development. So I'll start on the next slide, which gives just a little bit more of a linear workflow on what happens when the work comes into a biolab. Now most biolabs have the same way of working, where the project, once we have the materials procured, will be assigned to the research and development department, and they would be responsible for setting the assay up. So they would, with your key scientists, develop the assay from scratch, or would be responsible for transferring the method in from your lab or from another lab. We would evaluate any preclinical assay information, as sometimes we can garner some really valuable information about your molecule from how it behaved in the toxicology studies, and then we set the method up. And so depending on the complexity of the method, it can take anywhere from 2 to 4 weeks for us to set it up. In a stepwise fashion, we work through the bioanalytical guidance documents and the ICH-10 recommendations, just to ensure that you as a customer have a quality product that you can move through to your later phase studies in a seamless manner, and that the assay will be sort of future-proof. Once we have the assay set up, we write it up as an analytical laboratory method that gets handed over to our validation team, who then prepares all of the documentation in order to create a regulated bioanalytical project. So the project plan to be audited by a QA department, all of the analytical laboratory experiments will be carried out, audited, and that validated method would then be deployed on sample analysis at the start of the clinical study. All of this would be managed by a study director, which is a senior scientist within the team that is responsible for the conduct and the quality of the study. We put a data transfer plan in place with the CRO to ensure that we can seamlessly transfer data in a de-identified manner. And this is really critical for single and multiple escalation dose studies where fast turnaround time is needed in order to get the study progressed through its timeline, and enable the CRO and phase 1 unit to really adapt the design if needed to deal with the pharmacokinetic data that's generated throughout the course of the trial. So in the next couple of slides, just delve a little bit more deeply into Agilex Biolabs, as we are the leading bioanalytical laboratory with over 20 years experience. But what I really want to do is just give a flavor of what we can do and how we can assist clients and how we actually fit into that ecosystem that I had described earlier on. So we're a regulated bioanalytical laboratory. We have two major service streams, an LC-MS tandem mass spectrometry bioanalytical department and an immunoassay and immunobiology department. And across both of these departments, we have a parallel workflow where we can set up a pharmacokinetic assay according to their bioanalytical guidances and validate them for deployment onto the sample analysis. And then across both departments, we can measure biomarkers in a variety of matrices. And over the past 20 years, our scientists have seen most matrices, most novel chemical entities, and most biological entities, and they have a high level of experience in being able to develop and validate complex methodologies. We pride ourselves in timeliness and speed, and critically for many companies, we are registered for the RM tax. So we provide an end-to-end solution for regulated bioanalysis. I've listed some of the service streams here on this slide, from PK assays, immunogenicity assays for biologics, which we do in a tiered fashion. So we can do a screening, confirmatory titer, and neutralizing antibody assay for a variety of biological materials. And most of what we do are custom builds from scratch. So we work very exclusively in the early phase where it's novel biological entities and novel chemical entities coming through onto market for a variety of disease indications, from immuno-oncology to orphan diseases. But we also can support drugs that are already on the market where they might be repurposed or biosimilars. Pharmacodynamic assays, we have a very experienced pharmacodynamic immunobiology department that can set up immunology assays via multicolor flow cytometry in PBMCs or in whole blood, in order to assist the client in identifying exactly how their molecule is behaving biologically within the body once it's dosed. And we can also do assays such as cytokine release assays in order to determine any kind of cytokine storm or inflammatory reactions that might occur in the clinical trial. Where we don't currently offer service streams through global partnerships, we can provide a full service solution. So be it vaccine studies or virology studies, we can partner with global laboratories in order to ensure that those assays that are not critical to the fast turnaround, such as hemoglobination assays, neutralizing antibody assays that might be done towards the end, can still be contracted to a third party to enable us to do the fast turnaround front-end assay development and validation. So, as I mentioned, we work with novel chemical entities, novel biological entities, from proteins to peptides to conjugates to antibodies, to a variety of bispecific molecules and their various combinations thereof. Neutralizing antibodies. We work in information immunology, and we can set up assays for target engagement using our high throughput 20-color BD Symphony A3 20-color flow cytometer, and a variety of specialty immunological assays that we can set up in order to support immuno-oncology studies where these assays might be required. And then biomarkers that might be assessed either as an exploratory endpoint or as a fully regulated secondary endpoint throughout the trial. We can set these up to the latest global standards using best practice according to the latest white papers on biomarker analysis that have come out of the European Bioanalysis Forum and the recent issues in bioanalysis, the WRIB white papers, for example, where we attend these meetings on an annual basis in order to ensure that the level of service that we're providing is the highest quality to ensure success for the clinical trial. So I hope that this short webinar has just given you a little snapshot of various referral funnels that are available to bring your work into Australia. And that I hope that I've been able to highlight the criticality of getting the regulated bioanalysis set up ahead of the clinical trial. Sometimes this tends to be an afterthought, but really in order to take these assays off the critical path, we need up to 12 weeks to be able to get them set up, validated, get the long-term stability started ahead of first patient, first dose. And we want to enable success of the clinical trial and just really de-stress the client to ensure that those assays are ready to go before the trial commences. It's all about partnership and collaboration, and we'd be happy to engage with you in order to assist you in whichever way we can. And I'll thank you and leave it at that, and happy to take some questions.
All right, thank you so much. Thank you for that, Kurt. Actually, let's get started. Can you tell us what, in your eyes, is the biggest risk in a clinical trial, Kurt?
So the biggest risk in the clinical trial is really just, from a regulated bioanalysis point of view, is not getting the assays set up with proven stability ahead of first patient, first dose. So we want to ensure that when that sample is collected from the first patient cohort and it's frozen and it's stored at -20 because a phase 1 unit might not have a -80, that sample isn't in any way compromised. And so critical really to have that proven long-term stability, just to provide that reassurance that once the sample is collected, it's good to stay in that freezer until we batch analyze it for the clinical trial. And this is specific for trials where it might be a patient population, recruitment might be slow because it's a rare indication, and we just, the phase 1 units can only find so many of these patients in order to dose. And so it means that over time, the PK samples are stored in the freezer for batch analysis once they get to a certain number. And what we really want to ensure is that those samples, the integrity is not compromised. And so getting the regulated bioanalysis set up ahead of time and getting us that proven stability ensures that that the collection procedure is appropriate and the storage procedure is appropriate. I would say even more of a critical nature for many projects that have both plasma and urine, for example, where the drug might be sticky, it might stick to the tubing. You know, if we haven't done these investigative studies during the method setup, development, and then validation to inform the clinical protocol, the sample might be collected in a vessel that compromises the actual integrity of the sample. And so these are things that we evaluate during method setup to ensure that the lab manual is appropriate for the actual clinical trial. And again, it's all about just removing all of the risks associated with the trial and ensuring that the data that's generated, there's not going to be any questions by the regulators around the processes.
Okay, very well. Let's bring in Julia Jones, VP of Business Development, into the discussion. So Julia, tell the viewers here, in the client engagement process, so what stage should sponsors be speaking to the biolab scientist?
I think as early as possible. In fact, before they even speak to the CRO. And that's the opportunity to discuss the nitty-gritties of their needs, which are often very complex. As Kurt mentioned earlier, most of the compounds we work on are novel, so there's usually nothing off the shelf. So there needs to be a lot of intellectual sort of discussion that goes on right at the front end of them choosing to have other providers come in and help them. So as early as possible is my thought anyway.
Okay, well, so how quickly can you set up a scientific call, and who is going to be on the call?
So this is generally a common thread with our clients, and it's time is of the essence. Once they've decided to sort of start looking at where they're going to run their clinical trial, they want it done very, very quickly. So having worked in the industry for a long time, I've learned that checking my emails very early in the morning and until fairly late at night is key. To see if there are any incoming requests, and then I actually really quickly. So we're generally able to schedule a call within 24 hours and sometimes sooner. It's really dependent on the client's location and their availability. I mean, oftentimes when I get an email, I'll quickly email them or ring them and sort of say, "Is now good enough?" And they're a bit surprised by my So on our side, I generally ask them questions about what's the large or small molecule, and then I will make sure that we have a senior scientist from the relevant department on the first call to ensure that first discussion is meaningful.
Wonderful. And so who all will be on the call?
Myself, because I'm the chatter, but always a senior scientist, so that, as I say, it's not just a vacuous discussion about time of day, etc. It's actually meaningful with one of the senior scientists. So mostly, if it's a company that's got a large molecule, I'll have Kurt on the call with me. And if it's a small molecule, I'll have somebody from the LCMS department. So it's a relevant discussion right up front.
Wonderful. Okay. Kurt, let's talk about biolabs study rescue. So how common is this? And generally, how quickly can you take a rescue on board?
This has become a lot more common over the last 12 months. Really, it's a consequence of the SARS-CoV pandemic that really has shut down phase 1 units globally. We've been relatively unscathed in Australia with regards to COVID. So it's been business as usual for us. And so small biotech companies that are in that burning rather than earning phase, if they can't continue with their study, it's dead money, and they're losing revenue essentially. And so we're actually finding that it's becoming more and more prevalent where the study has stalled completely in the US or Europe, and it's just causing a lot of pain and anxiety for the customer. And at that point, the decision to bring it over to Australia is sometimes a no-brainer. And what we want to do is to ensure that that transition is as seamless and stress-free as possible. So, once we actually have the test item and the specialty reagents in from our suppliers, we can get started immediately. We're the largest laboratory of our nature in Australia. We have considerable capacity to be able to start these projects up, as soon as we get the reagents in, and as I said, start that critical long-term stability program, to ensure that the data at the end of the trial, is of the highest quality and that everything is being done appropriately. So, pretty rapidly, but it does all depend on whether it's a small molecule, large molecule, and what specialty reagents we might need to get in the lab. If it's a small molecule, for example, and the client can ship us the test item and internal standard, within a couple of days, we can start getting the method set up in a couple of days. So it really is a very rapid process, and we will work with the customer, in order to ensure that we can start with a letter of intent to enable us to have time for the actual contracting process to take place. So we never want to let the contracting be a hindrance and actually be something that delays the study. We have a variety of mechanisms to ensure that we can get everything set up really quickly from the front end.
Mm-hmm.
Very well. Okay. Well, Julia, let's talk about a fun business development topic here, about proposals and quotes and costs. So, for example, we have a small biotech wanting a ballpark for a board meeting or an investor meeting. How quickly are you able to turn around this kind of information to a client?
Well, typically we can turn something like that around in 2 to 3 days. I always ask them when their next board meeting is going to be, how soon before that meeting they need to have the information so that we can actually give them as meaningful information as possible. So if we have a synopsis and a patient schedule, obviously the more detailed our quotes can be, but we can do things that are not on the back of an envelope, obviously. But if they don't have a synopsis ready yet, we can make some suggestions about, the number of cohorts, the number of patients per cohort, and put something together for them to at least be able to present to the board so that they can, review that with them.
Very well. All right, Julia. So how about the last question for you, Julia? So which CROs do you work with?
Well, on one of the slides that Kurt presented, we did list a number of them, and they aren't all there because there wasn't enough room on the slide to put them all. But we generally have really good relationships with a number of great, or most of the great quality providers here in Australia and in the APAC region as well. And these partnerships have been developed over a number of years. We know most of the key players there very well. So it's a true partnership when we are dealing with them. And, there's different horses for different courses. Of course, each client needs a slightly different partnership across, depending on their needs. But we, in essence, can work with them all.
Very well. Okay. Well, thank you. I enjoyed that. That's all the time that we have for today, but we appreciate you spending time with us, and I'd especially like to thank you, Julia Jones and Dr. Kurt Sales, for your expertise and time, from Agilex Biolabs for sponsoring the discussion, and for Endpoints News, I'm Arslan Arif, and we will see you next time.
Thank you.
Thank you. Thanks, Arslan. Bye-bye, everyone.